Hi and welcome to this second video about genes and genetic testing, and today we're going to look at some more exam questions. So like before, these exam questions are taken from the Edexcel papers for the Edexcel AS Biology, and this is Unit 1. So the question is, thalassemia is the name of a group of inherited blood disorders that affect the body's ability to produce haemoglobin in red blood cells. Red blood cells are produced in the bone marrow. Oxygen in the lungs binds to haemoglobin and is carried to the cells of the oxygen that can be used in respiration. Beta thalassemia is the result of a mutation in the gene coding for the beta chain of haemoglobin. If a person inherits the gene mutations from both parents, this person will show symptoms of the anemia and will require blood transfusions. Symptoms of anemia include tiredness and breathlessness. Use the information given above and your knowledge of gene mutation suggests why a person with beta thalassemia has symptoms of anemia. Okay, so first port of call, you have to explain why they cannot produce the beta chain of haemoglobin. So therefore, there's a mutation, or essentially a change in the base sequence of the genetic material. This obviously gives you a different primary structure of the amino acids, and therefore you can refer to the beta chain being the wrong shape or it not fitting, something of that nature, or referring to the tertiary shape of the beta chain not being correct. From there you can explain that obviously the haemoglobin will no longer bind to the oxygen, and so you're carrying less oxygen. From that point, therefore, there's less oxygen being supplied to the cells. Therefore, the cells can't respire as frequently because they haven't got as much oxygen, and therefore you exhibit breathlessness because what you're trying to do is ventilate as much as possible to get as much oxygen in as possible because your carrying capacity is lower. And finally, of course, obviously, there's going to be tiredness due to the lack of energy or ATP production from respiration. So part B. If the phenotypes of the parents are known, the probabilities of having a child with beta thalassemia, an unaffected child, or a child who is a carrier can be calculated. Complete the table below to show the results of these calculations. And so these are the gaps you have to fill in. So the first bit you have to fill in is when one parent is a carrier and the second parent is a carrier. So obviously, if they're carriers, it's caused by a recessive allele. And so what we can do is draw a Punnett square for this. And so our Punnett square for two carriers will be, so if these represent the gametes for the mother, the gametes for the father, these are the permutations. So the probability of having a child with beta thalassemia, so in this instance, is 1 out of 4. So it's this individual here, so 25%. The probability of having a child unaffected by it, that's this individual here, so again that's 25%, and therefore the probability of a carrier is therefore those two there, so therefore 50%. You've then got an unaffected individual and somebody who has beta thalassemia. So this is the unaffected individual here. So he's the only gametes he can give across are two big T's at this point. And the affected individual again must have the condition, so must have the genotype small t, small t. You may use different letters for this. And so if you look at this, the probability of having a child with beta thalassemia, none of the individuals here of these four have the beta thalassemia, so none, or 0%. The probability of having an unaffected child, again, because they're all carriers, none, and therefore the probability of a child who's a carrier is all of them, therefore 100%. Now the problem with gene therapy is the fact that it's not always effective over the long term, and so therefore this type of gene therapy requires repeated treatment, so you could also mention that here, and so it's only 4 of those for the mark. Okay, so the final question today, gene therapy could potentially be used to treat beta thalassemia. Suggest how gene therapy could be carried out to treat this disorder. And so gene therapy involves the insertion of the healthy gene, and so that's possibly where we start. So the reference of the use of the normal or healthy gene for specifically the beta chain or for the haemoglobin, because it gives you it in the question. If we flick back, because it tells you about there you go, it's beta thalassemia, and the beta chain of the haemoglobin, you can use that in there. Therefore, you reference how you get it, basically the introduction of that gene or allele into the cells, again to another mark. You can insert it into the bone marrow, and again, if you look back at the question, it mentions the fact that the there we go, the red blood cells are produced within the bone marrow, and so that's information from the question that you could use. Also, you could then therefore explain how you're going to get it into those cells, and so the use of a vector, and of course you could then explain the types of vectors you could use, whether that's a virus or a liposome. And then you can credit the reference of any appropriate delivery of the vector, so for example an injection into the bone marrow.